Thank you very much. So we've got a wonderful panel here today. The subject of our discussion on this panel will be building for personalized medicine and synthetic biology. So quick introductions. I'll introduce myself, Matthew Konneh, former pharmacologist, consultant. I've worked across drug development, digital health, and now within venture development, working with startups, as well as looking at opportunities for a small venture fund based out of Santa Clara in California. On my left, I have Ohan, Mike, and Robert. Could you introduce yourselves, please?
Sure. My name is Ohan, of course. I am a scientist by training. So I'm in the biotech industry. So I work for a company called Immunocore, and I'm in antibody therapeutics and TCR therapeutics. And my background spans, back home in Australia was, again, antibody therapeutics. And I work in three major disease areas. So working on live drug programs in oncology, autoimmune, and infectious disease. So just I'm happy to share my perspective from the therapeutic side of things. So excited to be here.
Hello, everyone. I'm Mike Gavrielides. I'm a technical lead and senior data architect at the Francis Crick Institute. Although my background is in molecular biology and bioinformatics, through my career, I was always involved in strategic planning and technical design of software and data systems. This is what I find very fascinating and creative for me. And I think this is something that biology lacking nowadays, because we have so much data generated, and big tech companies like Google and Meta and Netflix, they also generate a lot of data, but they have the right technologies to manage all that data, which is not true at this stage within the hospitals and the research. So yeah, these areas are really interesting to me, and how we can leverage all these emerging new technologies in IT world and use them in biology.
Thank you, Mike. Robert?
Good afternoon, everybody. My name is Robert Thong. I'm the co-founder and CEO of a startup called Multiomic Health. Previously, for about 30 over years, I used to work helping pharma companies and biotech companies improve the way they run their R&D and to do partnerships with each other. The company I founded 4.5 years ago, what we do is we try and understand different patient subpopulations that don't respond to existing drugs. We then devise new product concepts for both diagnostics and therapeutics, which we partner with the drug and diagnostic companies. How we do it is we work with real-world patient data. We look at longitudinal health records. We look at all the different omics for those patients, over time, and we use that to understand not just why they are different, but how to go about treating them and diagnosing them. This topic area, of course, is exactly in the sort of space that we're working in, so I'm very keen to participate in this discussion.
Thank you very much. And so let's get into it. First question, perhaps to Robert. Which recent advances in personalized medicine, tissue engineering, cell therapy, gene therapy are you most excited about? I mean, there are a lot going on, so I'm looking forward to your response.
Well, probably safest to talk mostly about personalized medicine, because that's the sort of space that I'm in. But in particular, we've had genomics for a long time, and that's become a very established technology. But this whole new world of multiomics, epigenetics, transcriptomics, metabolomics, microbiome, proteomics, that helps us understand a lot more complex diseases, particularly chronic diseases over time. And then if you couple that with AI, machine learning, generative AI, the ability to cope with huge amounts of data and to make sense of it, that really allows us to do something in this space which we could never imagine before. So I think that's really a sort of good starting point for this. Yeah.
Absolutely. Thank you very much. Mike?
So my expertise is not in that area, but I'll tell you what, anything relating to gene editing and genome editing really interests me, because this is the reason that I got into the biology field at first place, the changing all the genomes and manipulating DNA. And the fact that with synthetic biology now we can literally design systems from scratch and create circuits and inject them into life, it's really fascinating. And I'm also interested in to see all this from the perspective of philosophy, and how this will pan out, and how this will impact the human intervention in life in general, because I think it's quite it's important now, because you can change something that can have very traumatic implications in the next generation. So it's quite, the responsibility is quite
Absolutely.
high.
I think we discussed, perhaps didn't have enough time to get into that, but it is really fascinating, the philosophical aspect of,
Absolutely, absolutely. But also from data perspective, it's interesting to see how data-driven discovery will impact techniques like gene therapy and cell therapy, because I think we do have promising precision therapeutic therapies for diseases, like, for example, viral treatment for cancer. But the clinical efficacy is not very high because the complexity of biology is quite high. And you can have, for example, you can attack with viruses tumors, but the immune dynamics between the virus and the tumor is not always the same. So you need to personalize these treatments.
Yeah.
So with AI and these data technologies, we'll try to figure out what.
And we'll come on to the AI aspect a bit later, but that's fascinating. Ohan, as a tissue, as a protein engineer, I'm interested in your answer.
Yeah, I think for me, the advancement with the most impact right now for the short term. So traditionally, personalized medicine, we've seen approvals like Casgevy and Kymriah. So these are cell therapies. Unfortunately, they're really expensive. They're high intensity in terms of resource and manufacturing. It's actually quite hard to get reimbursed, more so in the UK than the US. So a recent advancement I think is going to create an improvement in the field is in vivo cell therapy. So traditionally with these treatments like Kymriah, the cells are extracted from the patient's blood, they are re-engineered, patient goes through depletion and they're put back in. But with in vivo, the technology allows the gene editing or the expression of chimeric antigen receptors on these target cells within the body. So I think that's going to push the price down, and it's going to open up the market to be, ironically, it started off as personalized medicine, but it may move towards being off-the-shelf medicine. But what I think is that this is a proof of concept, this technology is proof of concept such that if there are genome-specific diseases, then the mRNA payload can just match the sequence required for that specific patient. So it's going to open up that field and make it more cheaper.
Fantastic, really interesting. I mean, from my point of view, one of the technologies which I think is really interesting in terms of it's come a long way in the last 10 years is inducible pluripotent stem cells, which I think now there's some phase 3 studies using these stem cells in patients with infertility issues. So we've really come a long way. So let's move on. My second question to the panel. How are advances in genomics, transcriptomics, proteomics, and data analytics changing patient care models? Mike?
Yeah, I can start. I think, like Robert mentioned, we have all this plethora of different data sets now from different modalities. So back in the days, you would be narrowed to treat a disease for a specific, area. You would test like few biomarkers, maybe. But now we have this angles from different perspectives. You have metabolomics, you have proteomics, you have genomics, you have microbiome data. And the diseases are now looked from all this, holistic view. And the treatments, although previously it was more like reactive, you got sick and then you you find your treatment. Now it will be, I think, more proactive, and you'll be able to prevent things. And people, I think, will own their data because it becoming cheap. So everyone will go and sequence their DNA. You know, you will kind of have a lot of information about yourself, and your engagements with the hospitals will be more than before. Previously, you weren't much engaged. Like you would have like some disease, which works for the most of the population. It's like one size fits all. This is what you get when you're sick. But now it's more like, okay, this is what I look like. You talk with a doctor and you find something more, you know, tailored to yourself.
So you see a future where you bring your own data.
Exactly. Yeah. I think that would be, you know, but there are some challenges there with, you know, security management and all that. But I think that would be the future, where you have like a portal with your data, digital twins, something similar to that, right? Yeah. Yeah.
Fascinating. Ohan?
Yeah, I think you know, we've come a long way from genome sequencing. So we had Genomics England's 100,000 genome project. So you know, all this information is getting integrated into NHS, and it's allowing us to diagnose rare diseases and things like that. But for me, the biggest impact, I think, is cancer stratification and cancer profiling. So based on, so treatment responses vary quite drastically depending on certain mutation states that are observed in cancer. So an example is like BRAF mutations or HER2 mutations. And so what we're seeing as an example is if we think about BRAF mutations in melanoma and colorectal cancer, depending on the mutation status, oncologists will change the treatment option. So for melanoma, if you present with BRAF mutation, then more likely than not, BRAF inhibitors are going to be effective. But in the case of colorectal cancer, with a BRAF mutational status, you're going to need combination therapies. So I think when we get to this level of looking at genetic links and all that stuff, it provides insights for oncologists. And like another example is like it's elucidating risk factors. So if I go back to colorectal cancer, you know, patients who have a particular variant of the PIK3CA gene are actually more inclined or have a higher risk of developing colorectal cancer when taking aspirin. So it's these little nuances, these links, these relationships that we're able to start to figure out more so once we have, you know, the generation of genome sequencing, data integration, and all this wonderful things. So
fascinating. I punt that question to Robert, who is actively working in this field, right, with multiomics.
Well, building on what Ohan is saying, I mean, precision medicine is real and it's here already in cancer, and it's accelerating. You know, your oncologist today will look at your biopsy, your genetics, all kinds of other parameters to figure out which of the current treatments make more sense for you. I mean, we're not quite at personalized medicine in the true sense, but I'm sure we'll talk about that later on. But as far as figuring out you know, I walk into the clinic, I have some cancer diagnosis, people can have a much better idea which drugs are more likely to work for me. In fact, it saved my mother-in-law as well, right? So that was really good to see. I think 20 years ago, she would have been dead. But today, you know, it's actually, I would reckon maybe 2/3 of the people with cancer still have a good fighting chance compared to before. It's not a death sentence anymore. However, the real opportunity, which is still in its infancy, is all the chronic diseases, particularly age-related diseases. And what's really interesting or sad, if you like, about age-related diseases is it's not just your genetics that drives it, it's what you've put yourself through over the last 50, 60 years. So you're born, you have certain genetics, so you have certain predilections to certain diseases, fine. But then if you don't look after yourself, you have a certain diet, you don't take any exercise, you spend all your time watching telly, etc., you smoke, whatever, your body works very differently from what your genetics is. So the blueprint that you were born with is actually wrecked, and you're working in another completely different way. And so without all these new omics technologies, we'll never be able to figure out how your body's working today, because it's not what you're born with, it's what's happened 40, 50, 60 years hence. Now, the other problem now is you've got a huge amount of data. It was bad enough with genetics, but now you've got all these other omics as well. And then in chronic disease, you're not just looking at one time point. You know, cancer is a disease where you'll probably die if untreated within the next 2 years, but you could have diabetes and you could have your foot amputated 10 years after you were first diagnosed with diabetes, right? So you need to look at all that clinical data over time as well, and then all the omics data, how that changes over time as well. So without machine learning, AI, we'd have zero chance of figuring out any of this. I mean, all of us who were scientists, we worked on three dimensions, drawing charts, looking at trends. When you've got, I don't know, I mean, some of the patients we're researching, we've got 300 gigabytes of data per patient. How the hell are you ever going to figure this out without the AI? God knows. So we're still starting, but at least we have a fighting chance. And within 10 years, you'll see more precision medicines in the chronic space, for sure.
Thanks, Robert. And that really nicely dovetails into our next question. What are the main challenges in managing, integrating, and securing high-volume patient data? Mike?
Let's start. Yeah, I mean, the sheer amount of data that's generated, it's a problem itself. So much omics data, wearables, wearable devices, imaging, health records. It's mind-blowing how much data is generated. And I mean, the velocity is also quite high. It's really fast. I mean, these techniques are becoming cheaper and cheaper. So you can sequence more patients for every study that you're doing. You can sequence them multiple times during the duration of the study. At the Francis Crick Institute, we have 120 labs and 20 core facilities. So you can imagine we generate terabytes of data on an everyday basis, and it's quite chaotic. Yeah, I mean, a couple of weeks ago, I was talking with one of our scientists, and she had a project where she would go to the King's College, she would do the image acquisition, and then she would put all these images within a portable hard drive, and she would bring the portable hard drive to the Crick, which sounds a bit silly, but it's actually more efficient and faster than transferring all that terabytes of data over the wire. But then you end up your desk full of portable hard drives. This is not the best way to manage data. But this is what happens, and we don't have all this appropriate, as I mentioned, technology to manage these big amounts of data. But then it also comes to the variety of the different data sets, right? All that data is not uniform. It's so many different formats and different ontologies that everyone is using. And which brings us to the other big problem, it's the integration, right? Biology is a big system, so when you look into your data, ideally you want to look in your data like more a systemic way from different angles, and to integrate all that data is very challenging when you have all these different data types. It's very challenging to kind of make an umbrella, like one data type for data model for an umbrella that will have everything underneath. Yeah, it's very challenging. And this is what we're fighting at the Francis Crick on an everyday basis, how to create systems that everyone can use. Because sometimes we find ourselves where you try to please everyone, but you end up pleasing no one. Because everyone doing things their own way, hospitals doing things with their own way. There are some situations that look a bit trivial, but they're still problems. For example, managing IDs. Like one patient can be involved in multiple studies. Every hospital will manage their own systems and their own IDs, for example, identifying a patient. So a patient will end up with multiple IDs. And then this propagates to all the samples. And then, yeah, go figure out which sample to be going about patient, and then the data is generated. So you need to link all these things together. And you become with some ID chaos. We find ourselves always in these difficult scenarios. But yeah, obviously then the security and governance is also big topics where we need to be compliant with GDPR and HIPAA standards. Encryption of data, end-to-end encryption is quite challenging when it comes to big data files. Yeah, you need to make your sensitive data from patients secure enough, but also accessible easily accessible to prevent the scientists to take that data and put it somewhere they shouldn't put it just because it's easier for them, you know.
Absolutely. It's this 3V challenge, you know, volume, velocity, and variety. How are you dealing with this, Robert, at Multiomics?
With difficulty. I think we all have these challenges. You know, we do research. Starting point is, you know, patient health records. Nobody, you know, you, me, whatever, with a patient health record has 100% clean data record where there are no errors, because historically, you know, a doctor, a nurse, somebody typed it in, you know, whether they've been having a late night or they were rushing to watch the football or whatever. So there'll always be some random errors everywhere. So first you've got to clean it. Then you've got to say, oh, the standardization between the record over the last 5 years has changed, so how do you account for that? Then the lab test has changed, and the error bars for the lab tests has also changed, so you can't just assume that the data from lab test A and the data from lab test B over the last 10 years is actually comparable, because it's not. And all these sorts of things. I think the other interesting thing that we found is people's interpretation of the patient privacy law varies according to individual hospital, individual NHS trusts. Some people are more paranoid than others. And so you have to, like, you know, calm their lawyers down, bring your own data privacy consultant to say, it's okay. And then there are other people that actually accidentally try to send us the real stuff. And I said, no, you can't send us that. We're not allowed to see that. Don't do it. So, you know, you have a huge variety of that as well. So the practical realities is a bit messy right now.
We had cases where we always try to obfuscate any identification of patients, but clinicians would put the actual names of the patients on the slides, and then they would take a picture of the slide, and then you cannot control that, right?
Unbelievable. So, Ohan, at Immunocore, how do you handle this problem of, you know, huge amounts of data?
With difficulty. I think for us, you know, being in drug discovery and engineering, we try to, there is positive data and there is negative data. And I think we assign a lot of importance to negative data, right? Because it's just as important for us. So it's very difficult. We generate thousands of data points, obviously not as much as what you guys deal with, but just trying to make a sense of it and translate that to binding, translating to efficacy is very, very difficult. So when we talk about AI models and, you know, speeding up R&D cycles, yes, it speeds up cycles. You know, very easily we have the capabilities now to design in silico drugs that bind to target, right? But we're limited to drugs that bind to the target and elicit efficacy, because nature is very complicated. There are mechanistic principles to receptor triggering that eludes us. It's, you know, so our models are, you know, great in terms of binding, and so this is the data that we generate. A lot of, you know, binding data and all that stuff, but we're trying to make sense of it and translate that, and it's very difficult.
Interesting. It's, I hope you sort of got on top of that challenge, and but let's move on. Perhaps a question to Robert. How close are we to seeing true personalized medicine integrated into public healthcare systems?
Well, there's two parts to that question. What do we mean by true personalized medicine? Right? If you're a sci-fi addict,
N equals 1, perhaps.
Yeah, if it's N equals 1, we're a long, long way from even developing that, much less actually putting it into the public healthcare system. Yes, you could say in CAR-T and similar things like that, where you engineer something specifically for you, it's relevant. But it's going to be so expensive to do this on a mass market kind of basis. And then secondly, the whole regulatory process, I think we were speaking earlier on, it goes against that, because the regulatory agencies approve a drug, one at a time, on the assumption it will be used by millions of people. If you have N equals 1 drugs, that means you have theoretically, for one, you know, for any disease, 8 billion possible drugs, given the number of people on this planet, right? So how are they ever going to approve that? And how are they ever going to do a clinical trial to prove all the different efficacy and aspects? So I think that's going to be a challenge. what's more likely is, what I call precision medicine plus. Right now, you have in oncology an ability to identify which drugs might work better in which patients. That will also come to other diseases as well. But then, you and I, we have, when we reach a certain age, multiple diseases. And those drugs will interact with each other. So we'll eventually have to build AI systems that can make sense of all the different precision medicine things we've identified. And say, for a person, you or me in particular, I might need 5 different drugs when I'm 69, 70, whatever. Which are the right ones for which ones, right? I mean, that's still to come as well.
Do you have any comments on that, Ohan?
Yeah, just to jump in off the back of Robert. So I think the future of true personalized medicine is different for different modalities, right? So in the near future, we're going to have platforms like DNA, RNA, oligo, therapeutics, mRNA platforms being a lot closer because they are easier to scale, right? What we are a little bit further away for is this conventional biologics, and that's what Robert was telling on. So, getting an antibody approved is very, very difficult. But what I do see the stepping stone, right, is stratified biologics. So an example is Vertex Pharmaceuticals. So for cystic fibrosis, we've identified there's 4 mutations or 4 different variants to the CFTR gene. Yeah. Vertex have created a suite of molecules that a patient will be screened, and depending on their mutation status, Vertex can prescribe a certain drug for that particular patient group. So with disease stratification, patient stratification, we might see companies in the near term providing a portfolio of drugs as opposed to N equals 1 further down the line.
Fascinating. So slightly variation on that for Mike. How can AI and machine learning accelerate personalized treatment pathways? The masses of data that's out there.
Yeah, so I think, we slightly already touched the subject. Diseases are like complicated things. Like you need to look at the diseases from different angles. So AI is the only way to interrogate everything together, right? The concept itself is not very new, I think. You would have some data previously. You could do some statistical analysis before. But nowadays, we have so much data, and you cannot do the traditional statistical analysis with to all that data, right? So the only way we have now is to just feed all that data into AI model, and AI will figure out what we need to do more personalized approach.
Excellent. In the interest of time, just perhaps one more question. Obviously, there's a number of founders in the room, and so how should founders navigate partnerships with research institutions such as the Crick or pharmaceutical companies such as Immunocore?
I'm not in that area, so yeah, I don't know.
Expert on that.
We generally at the Crick we are quite active in that area. We want to engage people to to translate research. We have a lot of groups like translation group and VC-related interest groups that they organize events to do like networking among everyone. We have some fundings that's given to some of the ideas. But yeah, I'm not the best person to...
And with you at Immunocore, Ohan?
So I think partnerships are going to be key. There's no way you can do it on your own. Partnerships should be based on whether they can secure data banks, tissue banks, biobanks, because I believe that's where a lot of the progress is going to be made. So look, to get these systems, to get personalized medicine platforms into healthcare, I think you need the big players with big capabilities. So whether that's big pharma, whether that's governments, whether that's local hospitals, you need to be making these partnerships because you want translational information. If that requires you going to hospitals and say, can I please have tissue samples? That's one way to do it because you just can't do it on your own. You need partnerships.
Okay. Great, thanks. One additional question, in light of our discussions around personalized medicine, how may funding strategies differ, say, for personalized medicine relative to traditional medicine? Question for Robert.
I'm not sure whether it's actually any different anyway. I mean, many of you are investors. You all look at the fundamentals, right? What's the problem I'm solving? What's my solution? What's the market? How do I go to market? Is that all going to make sense? So from a funding standpoint, I'm not sure whether there's any huge difference. I mean, okay, maybe if you're a pre-seed company and you're focused on one disease, you might have, an angel who's, their mother had the disease or they are a patient or whatever, and they're more willing to give you money because of that. But that was always the case before anyway. But
was it the complexity in manufacturing, the eventual size of the market?
Yeah, I mean, but it's back to the basics as well, right? So if you have, maybe you have a technology that can make 50 million N equals 1 treatments, in which case you're back to a big market again, right? So it really depends what it is.
Any comments?
Yeah, I think, traditional medicines, investors will value based on, specific molecules. How deep is your pipeline? What's the target population? How many sales can you do from the drug? For me, I need to do more research, but when we're moving into the field of predictive medicine, how do you value that? How do you say, how do you go, this person is, if they use our technology, they're going to rely less on the healthcare system by X many years. How do you measure that? How do you measure that value? Is it cost save on healthcare per divided by prolonged life year?
That's sort of that value-based medicine that everybody's talking about.
Exactly. And I think, in the personalized medicine space, there needs to be, investors need to see, profit. We need to get better at using metrics to measure that.
In the interest of time, it perhaps takes one question from the audience.
Thank you, wonderful discussion. I'm Samir from Lighthouse Innovations and previous career in drug discovery. My question to the panel is, drug discovery is costly and full of dead ends. How do you mitigate that cost of dead ends? So for example, you've got, various projects and 8 out of 10 don't work. How do you manage that risk of the cost of those 8 projects which come to dead end? Do you see an opportunity for that data to be shared with the wider community of drug discovery and, in the concept of circularity, for example?
Question. I think we'll need a couple of hours to answer that question.
We could have a whole conference on this. I mean, one thing that we as our company is trying to do is, we're trying to identify which drug targets make most sense for which patient subpopulations. And we've just started a collaboration, confidential, with a large pharma to sort of check their compound library of all their failed projects, whether this might work with them. Because one of the things about precision medicine is also the reverse case. So you can make what the statisticians call a type 2 error as well. You have a perfectly good drug because it's trialed on the wrong patients in your clinical trials. It's sort of seen as a dead end. And yet that's a perfectly good drug for the right kind of patient. As well as the usual reasons for failure, which just doesn't work at all, obviously. But I think data sharing will be hard because for a lot of the pharma companies, the molecule structures in that data is part of their asset base. But I think the way we've been trying to approach it is like going the other way around, like find the need and then say who's got something that might work on it. Because then they're more willing to talk to you because otherwise they would have to throw it away anyway.
Excellent, thank you. Ohan?
Just more simply, from an R&D perspective, we are better at refining the number of molecules we put through that go deeper into preclinical testing. So with the use of in silico models and AI structure-guided design, we can test less and less because we're going to be a little bit more right? So from a deep R&D perspective, we're no longer testing 20 molecules, using resources on 20 molecules. We're now, using AI-assisted guidance to say, okay, these 20 are fine, but refine your resources to this 6.
On that note, we'll have to bring the panel to an end. Thank you very much, ladies and gentlemen. Thank you.
Thank you.